Okay, I think we can get started. Hello everyone, welcome to the special seminar of the NCI-funded Big Data for Cancer Research workshop. My name is Min Zhang. I'm professor of statistics and the associate director of data science at Purdue University Center for Cancer Research. So it's my great pleasure to introduce our speaker for today, Dr. Jill Barnholtz-Sloan. So in order to have Dr. Barnholtz-Sloan to have the most time with you, I'm going to keep my introduction brief and share just a few highlights. So Dr. Barnholtz-Sloan got her PhD in biostatistics from the University of Texas Health Science Center. So as a researcher, she's nationally and internationally recognized leader in cancer informatics and data science due to her extensive expertise in biostatistics, population genetics, and human genetics. And as an administrator, so she really brings data science to all research domains, especially cancer research. So there's something special I would like to share is that so she has a 17-year-old son, and he wrote a cookbook published on Amazon to raise money for cancer research. So especially to Senju and AACR, I'm sure you're all familiar with. So now please join me in welcoming Dr. Barnholtz-Sloan.
Thank you so much. So thanks to Dr. Zhang and the rest of the Big Data Training for Cancer Research team for having me today. I'm really, really thrilled to be here. And I'm going to go ahead and share my screen. Okay, can you see it in the correct presentation mode?
Yes.
Yes.
Perfect. So as Dr. Zhang said, thank you so much for that kind introduction. I am at the National Cancer Institute. I serve in two roles there as the Associate Director for Informatics and Data Science in the Center for Biomedical Informatics and IT, and also as a Senior Investigator in the Division of Cancer Epidemiology and Genetics, where I do brain tumor epidemiology research. I do not have any conflicts of interest to disclose, and I am a full-time paid employee of the NIH. And today I want to tell you a story about big data and what types of data we have available in repositories that we have at the NCI, and then thread that together with an example in brain tumors about how some of these data sets have been used to impact how we diagnose and prognose for brain tumors. So I'll start out by talking about the NCI Cancer Research Data Commons. If we think about the scientific data life cycle, where it starts with a scientific need and identifying a critical area, research applications come in, research is funded, and then data is generated, collected, and submitted and made accessible. That's really where the Cancer Research Data Commons, or the CRDC, comes in at that step. And you can see on the right-hand side here, just a high-level graphic of the little cylinders are the different repositories and the different types of data there. We have ways of accessing those electronically, including 3 cloud resources that allow you to access the data and compute on the data in a secure environment. And I will go into detail of all of these components for you so that you can gain a better, more detailed understanding about what are the elements of the Cancer Research Data Commons and how can I leverage them for some of my own research interests. So we hope to empower researchers by providing access to state-of-the-art visualization, analysis, and interoperability tools for multiple different types of cancer research data in a cloud-based computational environment. And this really allows us to make sure that we are preserving the long-term value of NCI-funded data assets, that we are improving access to data, submission of data, and interoperability, and that we're able to accelerate cancer research by allowing folks to have access to these data and analyze them in an integrative multimodal fashion. The cancer research data is built on a data commons framework, where it is a reusable and expandable framework with very specific core principles and structures. And it's also built in modular components, which makes it more nimble for us to be able to make modifications and continue to modernize. And we do have centralized authentication and authorization, centralized indexing of all the individuals included in the data sets. And also we are leveraging cloud-based resource infrastructure. Like I said, we do have cloud computing resources available, and these cloud resources allow access to large data sets without having to actually download them or move them. It allows access to secure workspaces, analysis tools, workflows, and pipelines that are already available through the cloud resource, but you can also bring your own data and your own tools to integrate in with the data sets that we have available. And so we have 3 partners, 3 different cloud resources, the Institute for Systems Biology, and this is based on Google Cloud, and it's really great for command line, Google BigQuery, and specialty database access. Broad's FireCloud, which is also on Google, is great for running production pipelines. And 7 Bridges, Cancer Genomics Cloud, which is based on Amazon Web Services, is great for non-technical user interface and visual displays. If you'd like to learn more other than the data, the information I'm giving you today about the Cancer Research Data Commons, you can visit our website at datacommons.cancer.gov, which provides you access to a high-level video overview, as well as details about all the repositories, the cloud resources, and how you access them. Just to give you a very quick snapshot about statistics and impact of the Cancer Research Data Commons so far. On the left-hand side, I'm showing you information about the amount of data available and how many downloads occur for each of the different current CRDC repositories. The GDC, the Genomic Data Commons, has been around the longest of all of our repositories and has over 65,000 users per month, which is really incredible. On the right-hand side, you can see what the use of our cloud resources is also very extensive. And I think particularly impressive is the amount of public tools and workflows, and especially user-created workflows that are available within the cloud resources. We have over 300 different studies and data collections that have been released through the CRDC and over 200 scientific peer-reviewed publications. So now I want to spend some time going into each of the data repositories and not just giving you a description about what's there, but providing you with some examples about how you can use the data that's there. So again, just to go through it very quickly, the repositories I'm going to go over with you today are the ones that are currently in place, which are the Genomic Data Commons, the Proteomic Data Commons, the Imaging Data Commons, the Integrated Canine Data Commons, the Cancer Data Service, and a new data commons that's coming online as we speak, the Clinical Trial Data Commons. So I will spend some time now stepping through each of these different repositories, explaining to you what's available there and how they can be used to give you some ideas, hopefully, for your own research. So let's start with the Genomic Data Commons. One really great thing about the Genomic Data Commons is all the data that's available within the Genomic Data Commons is harmonized to the same genome standard and variant calling pipelines. So here are some of the data resources that are available in the Genomic Data Commons. What I want to point out specifically is the Cancer Genome Atlas data is all here. And that sort of was the birth of the Genomic Data Commons was knowing that we had a need to make the Cancer Genome Atlas data available as widely as possible. Just to remind folks, in case you're not familiar with the Cancer Genome Atlas, it includes over like 20 to 30 different types of cancer. And with the overarching goal of fully molecularly characterizing these cancers and making the data available. And these are the different types of data that are available through the Cancer Genome Atlas. Just a high-level infographic. There were over 20 collaborating institutions. There are over 2.5 petabytes of data available. And I'm sorry, there are 33 different tumor types, including 10 rare cancers, over 10,000 patients, and 7 different types of data that have been collected through the Cancer Genome Atlas. And this is just a high-level schematic showing you of the massive impact that the Cancer Genome Atlas has had on our knowledge about cancer. And the Cancer Genome Atlas started in 2006, but you can see that the publications continue to come out with important findings. If we think about how the GDC data can be used, just to give you some examples of how you can use it and interface with it for your own research, you can actually identify both high and low frequency cancer drivers. So common cancer mutations as well as uncommon cancer mutations. You can look at genomic determinants of response to therapy. You can identify custom cohorts of individuals and then compare those cohorts directly by mutation frequency and other clinical features. In addition, you can generate all kinds of plots. One of the plots that is sort of well used through the GDC is something called an Onco Grid, which is shown here on your left, which shows you across a cohort of patients and for a list of genes exactly what type of mutation is occurring and at what frequency across that cohort of patients. In addition, you are also able to do survival analyses based on very specific demographic and clinical features, as well as these genomic features. This is just giving you some high-level statistics about the GDC. Some of these I already showed you, but I think what's really interesting to show you is that as time has gone on your upper left, you see that the amount of data continues to grow that's released year by year, and that these data are actually used, highly used internationally. There are many ways that you can engage with the Genomic Data Commons. There are regular webinars. There's a listserv that you can sign up for. There's a help desk that you can contact if you need help with accessing the Genomic Data Commons data, or you have questions, and they also have a very active Twitter account. Moving on now to the Proteomic Data Commons, which also has been harmonized to a uniform pipeline across all of the data that's available. And I'll talk a little bit about what types of data are available there and how you might be able to access it for some of your own research questions. Just some metrics. Right now, there's 29 terabytes of data from 15 different disease types and 12 primary sites. The graphic here on your right shows you which primary cancer sites are available there. And it is one of the newer repositories, but it also is growing in size and international accessibility for its user community. The data that are included here are really take it to sort of to the next step beyond just genomic sequencing data. And you can see here that it has whole proteome, phosphoproteome, acetylome, glycoproteome, and ubiquitolome data available. And the data are available in multiple different formats. And a lot of the data that it's available here stems from the Clinical Proteomic Tumor Analysis Consortium, or CPTAC. So they also have a web portal similar to the GDC with a dashboard of available data organized by primary site and disease type. It has a very nice menu bar for navigation, for browsing the data, creating cohorts, and exploring the data. It allows you to look at a summary of all the studies that are available, to also filter by general information, biospecimen information, clinical information, and what type of omic data is generated. And it allows you to also search by identifiers and also by gene and protein names. It allows you to look specifically at study summary data so that you can better understand for that particular cancer type, where did it come from? How did the protocol work? What was the experimental design? How was the data collected? What type of data was made available? And what are the primary references for that data set? It allows you to identify a cohort of patients through a case summary ability, and then look further into their demographic, diagnosis, exposure, and follow-up information. It also allows you to look at sort of an Onco Grid-ish type of analysis using various filters to filter by different genes and aberrations on that gene, as well as clinical data and biospecimen information. There are several application programming interfaces or APIs that allows you to access and query the data from a more, computing type interface. And here's some more information that's available for you here on that. It also allows you to explore the quantitation data, and it can be visualized through a very versatile sort of heat map-ish type visualization analysis software directly with the PDC interface. And it allows you to do tracks at the top here that have to do with various clinical annotations and other sorts of histopathological annotations. I'll move on now to talk a little bit about the imaging data commons, where our goal is to share, analyze, and visualize de-identified multimodal imaging data, where we are using DICOM as the standard for all of the imaging files that are available. This just gives you a graphical visualization of how much data we've been able to release through the imaging data commons over the last 2 years. You can see that it's grown tremendously and continues to grow. And a lot of the imaging data that's currently available is coming from the CP-TAC studies and the TCGA studies. So it also has a user portal that allows you to interface with the data and look at what types of data are available by cancer site. It would also allow you to search for a particular type of imaging data, and then you'd be able to see across all cancers what's available. And it's really using metadata as the standard, allowing for this extensive search, as are the other repositories that I've already described. It allows you to explore the relevant data available. And so if there is radiological imaging data, as well as whole slide imaging from pathology, it's going to allow you to search and look at those data and to really understand the heterogeneity after allowing you, just like the other repositories, to build a cohort of patients that you can then explore further through the Imaging Data Commons interface. In addition, we are hoping that the Imaging Data Commons is setting up a really nice AI, artificial intelligence, machine learning workflow by allowing for fast data retrieval, visualization, and then you can use the cloud resources to help set up your compute and analysis. I want to give you a couple of examples now of some interoperability, leveraging data from the Imaging Data Commons and linking that with the Genomic Data Commons and cloud resources. This first example is from Drs. Kante and Erickson from Mayo Clinic, where they were interested in using deep learning approaches on radiomics data to predict mutation status of gliomas, which is a very common type of malignant brain tumor, from preoperative MRI scans. So they looked in the Imaging Data Commons, they established their cohort of glioma patients, they obtained the preoperative MRI scan images, they then linked that with genomic data available on those same patients in the Genomic Data Commons, obtaining from there the mutation information and demographics. And then they leverage the Google Cloud platform to match up all of this data and develop and evaluate their radiomic prediction model. Here's another example from the Hansi lab, where they were looking to use AI approaches to predict non-small cell lung cancer patient 2-year survival. And so they were able to leverage publicly available data in the Imaging Data Commons in collaboration with data that they had from their institution in order to replicate their pre-processing pipeline and validate their models, all using the Imaging Data Commons resources and the cloud resources. In addition, for the Imaging Data Commons, we are setting up an imaging imaging de-identification pipeline in order to make data more quickly AI ready. And we're doing this in collaboration with Google and Deloitte, and hoping that this will allow for easier, sort of use of researchers to be able to make their data publicly available through the Imaging Data Commons. Move on now to talk about the integrated Canine Data Commons. This is very interesting because it's sharing data available from canine clinical trials. In case anyone didn't know, dogs are also, certain breeds of dogs have a proclivity to certain cancers. And it's especially now making available data from the pre-medical cancer immunology network canine clinical trials and the comparative oncology program, which also includes human information. And so this is one of our newer repositories. This gives you an idea of how much data is there. It includes whole exome, whole genome, RNA-seq, DNA methylation, and clinical data available on each of the animals, linked with human data. And if we talk about a scientific use case, if we're looking at canines with urinary bladder cancer, and you say, okay, I would like to study dogs with urinary bladder cancer that have samples stored from a biobank, and I want to look at differential gene expression between particular stages of bladder cancer, I can leverage the integrated Canine Data Commons as well as one of the cloud resources, in this case, the Cancer Genomics Cloud from 7 Bridges, to do that analysis. And so if I start by looking at the integrative Canine Data Commons and searching for cases of urinary bladder cancer, and then I'm able to generate a manifest from there, so I know exactly which individual dogs and where the genomic files are. And then I can import that information up onto the cloud resources and create my data set and my workspace there, then allowing me to actually start to do, scroll through all of the files and make sure I have all the right files in order to begin my analysis and perform my analysis within the cloud resource without ever having to download my own data onto my own high performance computing resource. This is all through the Cancer Research Data Commons. If you're interested in engaging with the Integrative Canine Data Commons, here's their URL. They also have a help desk, and their point of contact is Gina. Talk a little bit now about the Cancer Data Service, which allows access to NCI-funded data that is currently not hosted by other of the repositories that I've already described to you. And it also includes some very interesting, I hope to this group, NCI-funded data sets. So data from other NCI-funded programs are available for secondary data sharing. I've already told you about a bunch of the data that's already available through the specific repositories in the Cancer Research Data Commons. And the Cancer Data Service allows for a little bit more of a higher security controlled environment to make accessible other data sets that are NCI funded and supported that may or may not fit within one of the other current repositories. So how do you know when you need to come to the Cancer Research Data Service, or sorry, the Cancer Data Service? It's really when you don't meet current criteria for submission to the other data repositories, and that maybe you have a new type of data available within your data set that does not fit with one of the other data types and one of the other repositories we've already talked about, and that you require additional storage and secondary data sharing capabilities that require a little more, special nuances. And so some of the data sets that we have available, you can find here on the Cancer Data Service website. Again, you can go to datacommons.cancer.gov, and you can look at a data browser. And by looking at the data browser, you can also upload your manifest into the Seven Bridges Cancer Genomics Cloud. And then you can look here to see what data is available through the Cancer Data Service. And you can import files specifically into this Cancer cloud and do your analysis right there. So this just gives you an idea. The Cancer Data Service was first conceptualized in 2018, so it hasn't been running that long. But as you can see, over the last 3 years, we've had a number of very large data sets and important NCI-funded initiatives that have put data here. And some of these data do require some additional steps to get access to them, but if you contact the Cancer Data Service team, they will help you navigate through that. I want to tell you a little bit about a repository that is currently started and will soon be available, which will share data from NCI-supported clinical trials. And it's going to be starting with making some of the match data, the molecular analysis for therapy choice data available, which is one of the first really large-scale NCI-funded precision medicine trials. And a precision medicine trial is meant to link a patient to a drug that targets specific molecular changes that they have in their tumor. And so you can see that our first release will come this fall, and we'll be increasing data over the next year, including more data coming from the NCI Match trial, from the NCI Moonshot Biobank, and also from the National Clinical Trials Network for other types of NCI-sponsored trials. And so here's where we currently are. And we are in the process of updating to a FSMA moderate status. So there will be some additional access steps, but the Clinical Trials Data Commons team will help you get through all of this. And so look out in the fall for our first release and website production. Here's some information about the current match arms that are being prepared and will soon be released. As you can see, these have all been recently published in the peer review literature through their primary publications. And so these are why these are teed up as some of the first sets that will be available. And you can see in the middle columns here exactly what drug was given and what the exact target was for that drug. And so in order to go on to this clinical trial, those individual patients had to have that particular target as a deficiency in their tumor in order to be eligible. So they are going to have lots more data available, and they are going to also leverage a CBio portal-like interface for ways to view and interact with the data. And they'll have their authorization and authentication that will fit with the rest of the Cancer Research Data Commons infrastructure. They also offer user support, outreach, and training. And you can email them. And as much as possible, they're going to work with and leverage the up-and-coming Data Coordination Center for the CRDC for training and outreach activities. So stay tuned on that. Talk a little bit about now, okay, so we've got all these repositories and there's so much going on, and hopefully you now have a good idea of what's in those repositories, what's available, how you might be able to use it to answer questions that you have for your own research. But what's really important is that we can use the information across these repositories, also called interoperability. So some of the challenges for data-driven research, which are really real, are there are inconsistent data sharing policies because there's really no universal standard for this, although the NIH data sharing policy will be changing in January of 2023. And I hope we'll be helping to standardize this for NIH-funded programs. The user skills and tools vary, across individual labs, across institutions. And I think that it's up to us through the Cancer Research Data Commons for us to make sure that these data sets are available to anybody, no matter what their background or skill level. And so we are working on some new interfaces for the data. And then, we have to make sure that the data storage and the data upload and usage is as easy as possible so that data sets can be combined without requiring multiple downloads, moving data, or multiple data use agreements. And then, of course, there's just complexity of data, right? As you start to integrate in all different other types of data, everything gets much more complicated. And so hopefully some of you listening today are interested in developing new analytical techniques for data integration. And I think that that's just a critical needed area. So we want to be able to discover relevant data sets across multiple resources using common standards. And this is where metadata really becomes very important. And we want to aggregate and analyze data across multiple resources, hoping that that will have maximal impact for cancer. And so we have a data standards service that is now working with the Cancer Research Data Commons that is coming up with a harmonized data model across all of the current repositories that I've just described to you that will allow for us to then aggregate across all of these repositories so that we can query and make it clear to everyone who wants to use the Cancer Research Data Commons and all of the data that we have available, what's there, what's available, so that you can then begin to understand better what's available so you can formulate your questions. We also have, and are moving towards a single sign-on across all of the NIH data resources through a researcher authentication service, or RAS. And this is sponsored by the NIH Office on Data Sharing. And it will be it's an NIH-wide effort that, of course, the Cancer Research Data Commons is also implementing. In addition, the Cancer Research Data Commons is one of such repositories. There are multiple other repositories available. You can see some of them here across the NIH. And everyone is moving to a cloud-based platform, hoping that that will allow us for a better interoperability and really creating a much bigger NIH-wide data ecosystem. So what are we thinking in the future for the Cancer Research Data Commons? We are working very hard to lower the barriers to data submission so that we can have a single submission pipeline. All different data types are accepted. And we're working towards an automated de-identification of images, as I described to you in my section on the imaging data commons. We'd like to make data usable and utilizable to everybody, no matter what your skill set is, no matter what your level is, no matter what your discipline is. And so we'll do that by offering a variety of tools and cloud-based portals to leverage, to visualize and leverage the data and compute on the data. We also want to make sure our infrastructure is robust, that it's modernized, it's sustainable and reusable. And we are developing comprehensive plans for this. And we want to make sure that for some of the sensitive data sets that we have all of the right security measures in place to protect those data. So as we think about the Cancer Research Data Commons really being the foundation for what we hope to be moving forward, the beginnings of a national cancer data ecosystem that holds within it or links to it surveillance data, patient participation data, discovery and analyses. Well, again, I'll just review the repositories that are available in the Cancer Research Data Commons, the Genomic Data Commons, Imaging Data Commons, Integrated Canine Data Commons, Proteomic Data Commons, the Cancer Data Service, and the soon-to-come Clinical Trials Data Commons. And so I hope that I've been able to get you excited about the Cancer Research Data Commons, what's available there, how you can start to think through how you might leverage these data for your own research. And we want you to come and use it, and we want you to ask questions. So please reach out to us. So that was a lot. Sorry, just doing a time check. And so I wanted to give a little context to how some of these big data sets really have already been used and continue to be used to have major impact for cancer. And because I do brain tumor research, I'm going to go ahead and give you an example of the impact on big data on brain tumors. So where are we in our knowledge about brain tumors? We have limited treatment options, unfortunately, especially in the targeted therapies or personalized medicine. We therefore lack individualized treatment plans. But we have, really limited knowledge about the biological basis of the disease. And we need to do better about risk factor measurements and, because we just don't have a single risk factor that accounts for a large number of brain tumors. We, as you will see, as I provide you this example in brain tumors, we are now incorporating molecular features of the tumor into more accurate diagnosis and prognosis. And we need to also be better in this space about integration of different types of data that hopefully will lend to us making novel discoveries. So just to put brain tumors in the right context for you, brain tumors come in two major flavors, malignant and non-malignant. These data that I'm showing you here from the Central Brain Tumor Registry of the United States, which represents 100% of the United States in collaboration with our partners at the National Program of Cancer Registries and the Surveillance Epidemiology and End Results Program. You can see that the vast majority of brain tumors in the United States are non-malignant, but probably the one that you're most familiar with is the most common type of malignant brain tumor, also called a glioblastoma. Brain tumors are highly heterogeneous, and there's many, many different types of brain tumors. So when you talk about brain tumors, you don't have to, it's important not just to think of malignant and non-malignant, but also what type of non-malignant tumor and what type of malignant tumor. Incidence of these tumors varies across the age spectrum. In adults, they are considered to be a pretty rare cancer. You can see these are incidence rates per 100,000. And in adults, malignant brain tumors are 8 per 100,000, non-malignant are 23 per 100,000. But in children, malignant tumors are much more common than non-malignant, and they are the #1 solid tumor type in children. So the blood cancers are the most common in children, but brain tumors are the most common type of solid tumor, especially for children under 5. But the incidence of brain tumors varies significantly by age and histology in the United States. The graphic on the left is showing you incidence per 100,000 for different malignant brain tumor types. So you can see if you just focus on the solid red line, you can see that glioblastomas are extremely uncommon until you get into the 6th and 7th decades of life. And on the right-hand side, showing you some of the common histologies in the non-malignant tumor category. And you can see by far that the most common type of non-malignant tumors are meningiomas and tumors of the pituitary. In addition, just like incidence varies by age and histology, so does survival. And so this is showing you by the 3 different age categories. So children, 0 to 14 years old at diagnosis, adolescent young adults, 15 to 39 years old at diagnosis, and adults 40 and older. The most common histologies for each of those groups. And these are Kaplan-Meier survival curves by histology. And you can see that, that it varies widely in terms of the survival by age and histology. Unfortunately, however, the red line here still denotes the glioblastomas, and you can see that by far they have the worst survival regardless of age. So how do we leverage some of these big molecular data sets, especially the Cancer Genome Atlas and the Clinical Proteomic Tumor Analysis Consortium? So the Cancer Genome Atlas is available in the Genomic Data Commons, CPTAC data is available in the Proteomic Data Commons to really heighten our understanding of the molecular basis of brain tumors and really impact brain tumor diagnosis. So we've known for a while about very specific individual biomarkers for brain and central nervous system tumors, and that they may or may not be, important for prognosis, potentially important for response to therapy prediction. But what the Cancer Genome Atlas data sets, so the glioblastoma data set and the lower grade glioma data set solidified for us is that these specific molecular markers are actually critical for the most accurate diagnosis, which is really important. So on the left-hand side, I'm showing you data from the second paper that was published on glioblastoma from TCGA. It included a little over 500 cases. And this is showing you a Kaplan-Meier plot that's stratified by gene expression subtypes. That in the original paper that was published that included about 200 cases from glioblastoma show that there definitely were survival differences between these gene expression based defined groups. Here you can see on this Kaplan-Meier curve that those 4 lines on the bottom, the solid purple, solid red, solid green, and solid blue, show that there's no difference between those gene expression based subtype groups, even though they may give us hints about biological differences. But there definitely is something different about the purple dash lined group at the top, which is called proneural GSIMP, which is a group of patients that have IDH mutations and a hypermethylation phenotype across the genome. And then this led us to the studies on the right-hand side in the lower grade glioma patients and comparing them with the glioblastomas, where we showed that the critical molecular markers for accurate diagnosis of lower grade glioma types versus glioblastoma are IDH mutation status and chromosomal loss of 1p and or 19q. And this all led to World Health Organization classification changes for these tumors in terms of diagnosis, which is really, really amazing that the TCGA data was leveraged in this way. And again, these TCGA data sets used here in these publications, which were diagnosis changing for brain tumors, are available through the Genomic Data Commons in the CRDC. So what was amazing about this is that for the first time, the World Health Organization in their brain and central nervous system tumor classifications, first published in 2016 and recently updated in the summer of 2022, are now not only talking about glioblastoma, but they're distinguishing between glioblastoma IDH wild type and glioblastoma IDH mutant. From this, the Central Brain Tumor Registry worked with our partners in the cancer registry community to enact a new molecular marker variable to collect all of these different critical molecular markers for the most accurate brain tumor diagnosis. And in 2018, that was the first year that these markers were reported for the United States. And here are some statistics just showing you what the landscape is of these tumors in the US. I think one important thing here is because this was the first year that these data were reported in the United States, there's still a fairly high proportion that have status unknown. And in the new data set that CB Trust just received, we will now have another reporting year, 2019, and we're hoping that the proportion of status unknown will decrease over time. And so what about the proteins, right? We talked about the proteomic data commons. I want to tell you a little bit about the glioblastoma studies from CPTAC that are available in the proteomic data commons. Why do we want to study proteins? Because they really are the workhorses of the cell. And proteins are the targets of many of these personalized medicine or precision medicine therapies. And meaningful changes at the protein level are not always present or easily discerned from genome level, genomic level information. So in the glioblastoma study, they aim to integrate proteomic and genomic information as well as metabolomic data. They used 99 brand new prospectively recruited glioblastomas, samples from those patients as well as clinical data, and 10 normal brain samples from the GTAC study. And they had a validation set also available, and then of course leverage the Cancer Genome Atlas cohort. And so these are all the types of data that were generated on that CPTAC data set. And they're in the process of generating a whole new set of data on a whole new prospective recruited group of patients for additional validation. But I think what was really interesting here is that there were some novel biological findings that without having the proteomic data integrated in with the genomic data, I don't think we would have been able to find. And we found that there was a signaling bottleneck in receptor tyrosine kinase altered tumors, which are really important because receptor tyrosine kinases are targets of many of these targeted therapies for precision medicine. So that's really important. We found that P53 protein abundance is regulated by protein and phosphoprotein effectors, which is critical because P53 is one of the most commonly mutated tumor suppressor genes in all cancers, including glioblastoma. We also were able to distinguish 4 immune subtypes that were characterized by very distinct immune cell population differences. And this could possibly lead to novel ways to stratify patients for additional therapies. And there were a couple other novel findings. And so stay tuned with this new data set that's being generated, whether or not we'll be able to validate some of these, we hope so. So this is definitely a team effort. I want to give special thanks to the CRDC and the CBIT teams. Special thanks to the Erickson, Conti, and Hansi labs for allowing me to use their examples of how they use the imaging data commons for their own research. I strongly encourage you to take a look at the Data Commons website at datacommons.cancer.gov. In addition, I'd like to thank my team at the NCI, as well as the CB Trust team, which I have the pleasure of being co-scientific principal investigator with Dr. Ostrom at Duke. And here's my email. I'm also pretty easy to find on the internet. If you just Google my name, I guarantee you no one else has a last name Barnholtz-Sloan. And you can also find me on Twitter at this handle. And that's it. So I will stop sharing. And hopefully I didn't overwhelm everyone. That was a lot of information, but I'd be thrilled to take any questions. Thank you so much for your time and attention.
Thank you so much. We have a number of positive comments about your presentation in the chat and also some questions. So one question is, so are we able to expand the kind of comparative studies to other animals besides dogs? For example, what about farm animals such as pigs that have significant organ similarities to humans, for example, the liver and the heart?
No, correct. I mean, that's a really, really good point. I think, so my understanding of the, kind of how the integrative canine data commons was born was that they had this immunotherapy network already running with these immuno-oncology trials in canines, where they would allow them to also have human comparators to that. You know, I agree with the person who made the comment that, you know, many times novel, you know, sort of phase 0 clinical trials, right, will be done in large animals first, right, to make sure that, you know, give us some idea about toxicity and tolerability. Pigs, of course, are included in that. And so I think if you have some ideas about other, you know, animal models, and if you know of any clinical trials, especially that are running in those animal models. So this is very interesting about the canine studies is it's a network of veterinarians that are running this clinical trial together, similar almost to how the like human cooperative groups run, you know, trials. And Dr. Toby Hecht at the NCI is the federal lead, you know, federal stakeholder for the Integrative Canine Data Commons. And I'm sure she'd love to, you know, talk more about other potential animal models if you know of those. But I agree. I think it provides us a foundation to then build from to expand to other animals.
And the same attendee said brain tumors in pigs and dogs are very similar. So
yes, there definitely are very specific dog breeds that are susceptible to brain tumors. Hopefully not my sweet puppy who's laying behind me here. He's being very good. He hasn't barked, so this is good. Yes, pigs too, interestingly enough. So
thank you. And then we have a couple questions in the Q&A, but also Santiago has his hand up. So can I allow him to speak now?
Absolutely.
Great. I'm going to click allow to talk. So Santiago, you can unmute.
Oh, thank you. I wasn't sure if that was working, so I was also typing my question. But thank you. That was a fabulous presentation, and I wanted to ask. So, I have accessed publicly available data from. I'm interested in multiple myeloma, and there's an MMRF Compass data set. They have their own portal. And I see that the GDC portal pulls in that data, and I was curious if if the data is being reanalyzed using a distinct, unique pipeline that's different from the one used in their portal, or if you could comment on that. Is the data being pulled in as raw data and then being reanalyzed with a pipeline?
Yeah, so I don't know about their portal and how that works. I can only speak to the Genomic Data Commons portal. So any of the data sets that are available through the Genomic Data Commons are pulled in as raw files, and they are rerun through, the QA/QC and calling algorithms that are standard across all the data sets that are available in the GDC.
I see. Thank you.
You're welcome.
And then let's see. So one question from the chat is, so let's see, great talk. And they're also very interested in the databases. Are there any databases that include metabolomics or microbiome data?
So all of the data sets available in the Proteomic Data Commons do include metabolomics, because those studies are doing kind of a comprehensive proteomics, phosphoproteomics, metabolomics, lipidomics, glycolomics, okay, all of those types of omics. And then they're also generating genomic data as well. So you can look at those types of data as well as the genomic data and clinical data all together, okay? In terms of microbiome, absolutely there are NIH-funded researchers who are generating microbiome data, but microbiome data, as far as I know, in the main repositories, so the GDC, PDC, ICDC, IDC, are not there, but there may be some data sets in the Cancer Data Service that include microbiome. And I'm sorry that I don't know the exact specifics there, but you can go to the browser tool for the Cancer Data Service and see if there's any microbiome data available there.
Great, thank you. And then we have another question. Let's see. Hi, thanks for the wonderful presentation. Other than cancer phenotypes, are there any toxicity related phenotypes in the data commons?
That's a good question. So it all depends on what sort of follow-up data was collected as part of the studies that are available. So some of the studies will collect toxicity and progression data, so that you can actually look at not just overall survival, but progression-free survival. But a lot of the studies that are older didn't collect a lot of that detailed information, I think because they were mostly just, it was so new to be generating all these different types of data together. They just want to make sure they were doing everything, in the right way. But some of the newer studies, like especially in CPTAC and some of the TCGA studies that are newer, and some of the other studies that are available that are on the newer side, probably do have more extensive information available. But for sure, in the clinical trials data commons data that will be made available, obviously as part, it's standard as part of clinical trials to collect those data, and those data hopefully are going to be available there for those clinical trials.
Thank you. And then let's see. Another attendee asked, could you describe in broad terms accessibility for non-US based researchers and also academic versus corporate users?
Good question. So you can access the data no matter who you are or where you are. The data are, freely available. Where some of the costs come in and maybe some of the additional steps you might have to take are some of the data sets are more secured for reasons based on how consents were obtained from the patients and how the data can be available and those types of things. And so you may have to go through some additional steps to get access to some of those more secured and controlled data sets. For sure, the data in aggregate and kind of more results are publicly available through the web. So you shouldn't have to there shouldn't be anything that precludes you from accessing no matter where you are in the world, except for maybe internet connection. In terms of the cloud resources, if you're interested in doing your own compute, they do offer you some free cloud credits to get you started. But then once you've used up those credits, that compute time and that storage space, you would have to pay your own costs on the cloud. So I hope that answers. Great. I'm not sure that distinguishes really between academic versus industry, by the way. So
no, I think academic users also have to pay for time on the cloud.
Yeah.
Unless they apply and get funds.
Right.
So
exactly. But to get you started on the cloud resources, you can start with some free certain number of hours of cloud credits. And some people are able to do the analysis they want to do within that boundary, right? And never have to pay additional cloud costs.
I think it's like $300 of cloud credits or something
Correct.
like that per researcher.
Yes, right. And you can also apply to get more than that.
Oh, I didn't know that.
Mm-hmm. Yeah.
No
guaranteed, right?
Yeah.
But you might as well apply. Yeah.
And then let's see, is germline mutational data available via the GDC portal, or is that just somatic mutations? And if both are available, are different pipelines used?
Good question. So the through the portal, they're showing just the somatic mutations, and those somatic mutations are generated from algorithms that do compare the tumor genome to the matched normal genome for each individual. Okay, so they truly are somatic calls. And you can look more into the documentation on the GDC pipeline, but they use pretty well-established callers for that. And they use, I think, 3 or 4 different callers, and then they take the consensus call. So you can get access to the germline data, but that requires additional steps and additional review of access in your project and all of that. But you can get access to the germline data if you're interested. The type of germline data that's available, by the way, sorry if I might, Nadia, varies depending on which cancer type it is. Okay, so for some of the cancer types, it would be adjacent normal tissue, which may or may not be normal, by the way, just be careful, and also blood. All right, for brain tumors, can't really say, hey, gee, could we take a piece of your normal adjacent brain, right? So for the sorry, Min, am I making you laugh? For the brain tumor studies for glioblastoma and lower grade glioma, the normal is blood from the individuals. Sometimes the GTEx samples are used because those are coming from normal tissues. Yeah.
Oh, there was a question from Andrea in the chat.
Okay.
So the question is, many publications use NHANES data and no additional data. So does this also happen with NCI stored data, or do the studies utilizing the NCI data typically include additional data? Thanks for a clear talk.
You're welcome. Yeah, so the NHANES data is completely different type of data, right? The NHANES data is from surveys over time on individuals looking at specific risk factors and lifestyle features for other types of complex diseases, including cancer, you know, but there's no omic data there, just to clarify there for the rest of the audience. You know, I think it would be hard to make a general statement about how is the data used. I think it's used in a few different ways, right? One is that you kind of have an idea, or you have a favorite gene or a favorite protein. And so you go in and you use the browsers that are available just to sort of query the data and see what might be out there, and to see if your hypothesis is even worth something, right? You know, would there even be a large enough number of individuals with that specific aberration in that specific gene that you're interested in, okay? The second is that you have your own data set from your institution, and you've generated some results, and you now need an independent validation set. And so the data sets in CRDC are leveraged a lot as independent validation sets. The third would be the other way around, right? You leverage the Cancer Research Data Commons data sets to do your discovery, and then you have your own institutional data set for independent validation. So I would say those are really the 3 big ways it's used. And I think when I talked about sort of the future of making sure that our interfaces and data were usable to anyone, no matter what your discipline, what your skill level, what your sort of level and rank is, we're hoping that really impacts that first example, right, that I gave, which is anybody interested in cancer can come and query the data and ask questions, and maybe help just even determine whether or not their hypotheses are feasible, you know.
Thank you. Yeah, there's another question, I think 2 questions from Gamel. The first one is probably when we talk about the cost, like what is the average cost per year? And the second one is, are we planning on adding spatial biology data from histological samples from Akoya, 10x Genomics, etc.?
Okay, so maybe you can clarify for me when you say what's the cost per year? I'm not sure what cost you're asking for. Do you want to know how much does the Cancer Research Data Commons cost, or do you want to know, oh, yes. Okay. Well, it costs a lot of money. So, and we really have two sources of money, right? We get money through the regular NIH budget to help support the Cancer Research Data Commons. And then we also are receiving money from the Moonshot because one of the Blue Ribbon panel recommendations was developing a cancer data ecosystem. And so we received 2 pots of money, and it's a lot. It's a lot. It takes a lot for us to make sure that all of the data are compliant and that they are available. And you know, we are in the process of doing a sustainability study for the Cancer Research Data Commons, and we're really, with some help from Deloitte, we are taking a really, really hard and close, detailed look at how we are spending the money to make sure that we are spending it in the best way to make the data as available and accessible and interoperable as possible. Sorry, the second question?
It's about, are we planning to add spatial biology data?
Yes. So one of the data sets that's coming in through the Cancer Data Service, and the pieces of the data will be available through the Cancer Research Data Commons repositories, is the Human Tumor Atlas Network, or HTAN. I'm not sure if you're familiar with that, but it's really meant to be like a human tumor atlas. And so available with those data will be spatial transcriptomic data and also kind of multi-layered imaging data. I'm not sure if this is what you're asking for. I should say available currently in the imaging data commons are CAT scans and MRIs in DICOM format, as well as whole slide imaging from the H&E tumor sections for those cancers, also in the DICOM format. So you could do a spatial analysis of those, right? Don't ask me what algorithm to use. That's not, I can bring you to the right people to talk about with that if you'd like.
Thank you. Yeah, that's a lot of information already. And I just realized, you know, Gamal also asked a question early on, very early on in the chat, that new tutorials are needed for users to learn how to exploit these resources.
Yes.
So I don't know if you, like, there's any tutorials available? Yes, they are definitely working on those, and we recognize that that's something that we really need to do. And we are developing a very detailed, you know, we're calling it a communications plan, right? But training obviously is really important. Someone wanted to see the dog, so let me see if I can get him. There he is. He's sleeping. He kind of blends in. He's a chocolate lab. Duke, come here. No, he won't come. But he has his pigeon there, which he carries around with him, which is oh, here he comes. Hey, did you wake up? He thought my lecture was very, very boring, by the way, just so everyone knows. There he is. His name is Duke. He's a 4-year-old chocolate lab. So, and ironically, he was bred from bird hunting dogs. That's the breeder we got him from. And then he latched onto this pigeon, this stuffed pigeon that he carries around with him. So he obviously is not a bird hunting dog, right? So I do data science, not bird hunting. So. Yeah. I think, Gamal, do you want to talk? You are free to.
Yes. Can you hear me well?
Yeah, we can hear you.
Very well.
Yes, we can hear you.
Jill, I really thank you very much for the wonderful presentation.
Thank you, Gamal.
That's very enlightening. But I have a question, Jill. Have you heard of the Human Cell Atlas?
Yes.
Okay, so right now, all our cohorts and all our data is associated with cancer. But if we were to have a mirror and we were to put together the Human Cell Atlas as a mirror to our database, so that we can have a baseline for comparison. So most of the time, what we do is a snapshot of a movie, and we look at it as if it was the cancer itself. But in reality, cancer is a spatiotemporal phenomena. So how do you envision the possibility to collaborate with the Human Cell Atlas and put together the data that is being collected in the Human Cell Atlas as a mirror to the databases that we have for cancer?
Right. No, it's a great point. And I think, there are other resources available that are mapping at the single cell level, right, of normal tissue. So you think about like the Allen Brain Atlas, and there are other resources like that. That's where the interoperability and working together to try to organize data per the same standards, and where having the right metadata really comes into play, because that does allow you to search across and interoperate between these repositories. And I know I only showed one slide, but the Cancer Research Data Commons is part of a larger data ecosystem throughout the NIH that includes all these other repositories that are being developed, including one, there are repositories where the Human Cell Atlas data will be available. So hopefully, there's going to come a time very soon where you would be able to grab data from CRDC and grab data from these other repositories. You can do it some now with some repositories and analyze all of them together. And hopefully you would be able to do that in one of these cloud-based environments so that you're not downloading like petabytes and petabytes of data and having to pay, those download charges, which, as you can imagine, can become pretty expensive very quickly.
And the other question I was going to ask you, Jill, is that when we could try to correlate from multiple patients in the imaging data side, we do not have a coordinate framework that is being like what the Human Cell Atlas and the HUMAP are trying to establish. Because I get really lost when I have a cohort of patients, let's say for brain tumors, but the brain tumors are all over the place, in terms of their location. You can say, okay, it's a GBM, but say GBM where and how? It's frontal. Well, it's in the frontal lobe, but where within the frontal lobe, etc. A coordinate system is called a common coordinate framework is what is being proposed by this Human Cell Atlas and the HUPMAP consortium. Will it be possible to do something like that within our imaging databases?
I mean, I think that's a wonderful suggestion, Gamal. I agree with you that it's very easy to get lost in the image without having some kind of, reference point. I would strongly encourage you to email the IDC team and set up a time to talk with them about that idea and find out from them what they're thinking in terms of that. I think it's a really important point.
Thank you, Jill.
Thank you.
Yeah. Thank you. Yeah, actually, I have a quick question, if you don't mind, if I have a few minutes.
Sure.
Yeah, so I think, these are really great, data resources, especially for us, like statisticians. And I think somewhere you mentioned that, the need the tools for more like integrative data analysis. So I'm wondering if you could elaborate this a little bit.
Well, I mean, I guess, from a statistical standpoint, right? Really what it's about is, how do we subset patients, right, into clinically meaningful subsets? And I think you can use each of the different types of data to do that. But then if you start to integrate all this information, you'll find that your subgroups of patients changes, right? And so, a lot of there aren't as many techniques available for integrating all of this together as there are available for any single type of data. right? Like if you think about exome sequencing or RNA sequencing data analysis, there are established standards for calling mutations from exome sequencing data and for doing differential gene expression analysis from RNA-seq data, right? But what, I would challenge all of you guys, since you're all in the big data space and you're, building careers or maybe changing careers in this space, to really think about that next horizon of, okay, ridiculous amounts of data are now available. What do I do with this? How do I make sense of it? A lot of people are using a lot of novel approaches to visualize the data, to cluster the data, to, graphically represent the data. So I mean, to me, that's what I think moving forward, that's really where the really interesting results are going to come from, because as part of clinical care, many cancer patients are, their physicians are running all these assays on their tumors. So the data are going to become more and more available. And, how do we take it from like research generation of data, to really using it in a clinical translational sense to make a difference for patients? I mean, we've obviously made a big difference already using genomic mutation data, because we know that there are certain drugs that target certain mutations. And if we see that mutation in that patient's tumor, we can possibly use that drug that targets that mutation. But I don't think we know quite what to do with proteomic data or gene expression data or lipidomic data, right? Or microbiome data. So, I think it's going to require some really novel analytical thinking.
Okay, thank you. Yeah, that's very helpful.
Jill, can you elaborate on what you just mentioned on this specific connection between genomic, proteomics, lipidomics, what they call it, the other one is microbiome, the microbiome. Could you elaborate a little bit? It's on, you said that you don't envision yet the connection.
No. For sure, there's a connection, right? Because each of those different types of data are being generated from different pieces of the biology of the individual and their tumor, right? So absolutely, there's a connection. I guess perhaps I misspoke, and I apologize for that. I guess what I mean is like, how do we make sense of all of that? I think is the hard part, So if we get a signal at the DNA level, the RNA level, the protein level, the lipid level, how do we analytically make sense of all of that information so that we can help a patient? I mean, I think that's what we all want, right? We all want to make a difference for individuals with cancer, so how do we think about that, like, integration of all of that information and what it's telling us about the biology of the disease?
The keyword you just mentioned, integration, that's the keyword. How would you integrate the data? Yeah, okay.
Right, but it's hard because remember that the signal you get at the DNA level, the RNA level, the protein level are not directly correlated, right? They're not perfectly correlated with each other. If they were perfectly correlated with each other, then you can say, well, as long as I have the genomic information, I know everything else about the biology of the tumor. But that's not the case at all.
See, there was a program at the NIH. It was called the Cancer Degradome. Many years ago, and there was a book written about that. And you're just totally correct, because DNA to RNA to protein doesn't mean that it's a straightforward path. Everything has a spatial domain and a half-life. A protein that is being produced instantly doesn't mean that it's going to survive for a long time. There is what they call process of ubiquitination. The proteins completely get destroyed as soon as they are produced. So maybe the gene is active, but that doesn't mean that the protein is there. You know, yeah, you're right. The integration is, I think, it's going to be a significant research area in the future. Yes.
I mean, the integration is the way to go, I'm telling you, because data is generated so rapidly. You know, most of it we don't know what to do with, right? It's just too much. So just because the technology, I think, is ahead of the analytics.
Yeah, there's always a kind of lab after
that. Of course.
Yeah. But just curious, is that where the like the NCI funding is also leading towards too, you know, in terms of the analytical tools?
Well, there are definitely mechanisms that you can apply for. The ITCR is one in particular. There are multiple different types of grant mechanisms you can apply for through there. That's the Information Technology for Cancer Research. And one of the big things there is to develop new algorithms. Absolutely.
Okay, thank you. Heather, I think we asked you so many questions already.
No, it's great.
And yeah, I know you have a meeting at 2:30 and you have a meeting before this and right after this. But yeah, thank you so much for the wonderful talk. And yeah, I guess we learned a lot of information. Yeah, I will definitely ask for your slides and video post the recording that, you know, more people can see it.
Yeah, my pleasure. I'm just thrilled that you asked me to be here, and I hope that everybody learned something they didn't know about before, and please come use the CRDC. Please, please, please. So.
Yeah, we will.
Okay.
Yeah.
Take care, everybody. Thank you so much. Bye-bye.
Bye.